Okay, guys. Ready? Yes? Okay. So welcome to our Winogradsky column class. I hope you all have fun, and I'll be guiding you through this session. So can you move to the next slide, please? This is our agenda for today. We're going to do a quick warm-up video, and the question will be based on the video. Then I'll talk to you a little bit about Winogradsky columns. Then we'll get into the demo. Then in our group work, I will give you a few questions to work on, which we essentially will be talking about in the class. And then if you guys have any other questions that you have, you can talk about them in the group, and then we bring it back and when we reconvene and we could all discuss it. And then we'll close. That's what today's looking like. Next, please. Okay, so this is the video. Can we start at 44 seconds? And we'll stop at 5 minutes, 44 seconds.
Can you guys see?
I don't think we can.
Sorry, what was the, you can't hear it?
No, sorry.
In your audio settings, there should be an option to play computer sound. Alejandro, do you remember where that is?
Yeah, so actually, if you like scroll to the top, you should see like a little pop-up menu for the share screen.
Mm-hmm.
If you, I think there's like three, an ellipsis. If you click on that, it should say share computer sound.
And it should be a small little checkbox.
Mm-hmm.
Okay, I think I got it. Let's try it again.
Soil life. Research shows that the widespread use of synthetic fertilizer leads to soils that no longer have the required biology for healthy plant growth. Fertilizer comes from a time in which there was no understanding of the long-term effects on the soil. For farmers, fertilizer was a miracle. The harvests doubled, and growers were able to provide the required food for a growing population. But now, after years of using synthetic fertilizers, the effects on soil quality are apparent. Increasingly, it is seen that fewer nutritional minerals remain in the soil, resulting in plants that quickly become weak and diseased. They are no longer protected by bacteria and fungi, but instead are attacked by them. As well as this, using fertilizer means that these days, farmers need to plow deeper as the ground becomes quickly compacted. This is a costly business for farmers and is disastrous for the soil. There is a better way. It is not widely known, but the roots of plants are able to develop more easily in an environment rich in old roots. Here you can see the current plowing method in which the soil is turned to 25 centimeters. This means that the plant really struggles to reach the thousands of passageways that other plants have created with their roots. They often don't even reach through the plow pan, which is caused by plowing the land with heavy machinery. A surface cultivation method can offer a solution to this. This shallow soil processing means that the roots are quickly able to access the passageways left by decomposing roots. In this situation, they only have to work really hard once they reach the extremities of the passageways. This means that they can root deeper year after year and ensures that there is even more soil and water available for growth. Dealing with diseases by spraying does little more than suppress symptoms. A healthy plant is able to keep most plant diseases out. In response to the negative effects of using fertilizers, pesticides are used to save the harvest. The conclusion we can draw is that plants that are only given fertilizer will ultimately always become diseased. Fertilizer is the direct cause of the use of most pesticides. In order to resolve the worldwide problem of over-fertilization, the vicious circle must be broken. The question that we must ask ourselves is, how can we improve the soil? The answer is surprisingly simple, but requires some explanation. Just like every other organism on the planet, a plant does not function independently. Health and growth depends on a partnership between specialist bacteria and fungi. Plants, fungi, and bacteria form a wonderful system based on trading products. How does it work? Fungi and bacteria supply nutrients from the soil that would otherwise be hard to reach. In exchange for this, plants supply sugars in the form of glucose. To explain this system, we will explain bacteria and fungi separately. Plants have a problem. Their fine absorption roots have a limited capacity. They only use 4 to 7% of the soil volume and are, on average, not thicker than a hair. They don't live long, only 1 to 3 weeks. As you can see, plants need external help. Plants use their absorption roots to forge a partnership. If they can't find partners, they'll die. If they succeed, it's the start of a beautiful relationship. From now on, a symbiosis can be created. Minerals are hard to mine from the soil, but rhizobacteria are specialized in this process, particularly when it comes to releasing phosphate, which is very important for the plant. These bacteria also carry out countless other important tasks, including one which is very special. Rhizobacteria form a sort of natural defense system around the roots. Anything attacking the plant is kept outside by the physical presence of these rhizobacteria. They ensure that there is no room for disease-causing bacteria. They have their own interest in this because the plant provides them with food. However, the bacteria also need help. They are not travelers. The bacterial colonies do not move back and forth from minerals to the roots.
Sorry, just checking. What was the stop time?
That's good. That's good.
Okay.
And slide, yeah. Okay. Then we'll go to slide 4. Yeah, there we go. So, guys, what do you think? What is your understanding of symbiosis from the video, and how does it benefit the organisms that were spoken about in the video? Anyone? And this is important because we're doing a Winogradsky column, and this is a self-sustaining ecosystem. And for any system to function, there has to be some sort of symbiosis, right? The participants of the system will have to depend on each other for the system to work. So that's why I kind of brought it in here. So the next slide, please. Okay, great. So one example of this that is used in agriculture is crop rotation. Well, I'm not sure if it's used today, but I know that it was used at some point. So crop rotation is exactly what it looks like. You're moving crops from one part of the land or from one plot to another. And why do we think that this might be beneficial? Why would we move one? Why would we plant a plant in a different location than we did before?
So I'm guessing, being as though if you have different crops, each type of crop would need like a certain amount of minerals from the soil. So if you switch one that takes a lot of minerals from the soil and switch it to an area where crops, you know, don't take as much, it would kind of let the previous plot kind of replenish itself.
Yeah, that's a good example. Exactly, right. So crop rotation, oh, Alejandro? Okay. So yeah, as Alejandro was saying, essentially we're keeping the soil fertilized. We're keeping the quality of the soil. We're keeping up the quality of the soil. So can we move to the next slide, please? Next slide, please. Okay. Okay, so this is the traditional crop rotation. These 4 plants are always used. Okay. Wait, no, previous, please. So we have leafy plants, we have fruits, we have roots, and legumes. So legumes are known for their symbiotic relationship with nitrogen-fixing bacteria. And nitrogen-fixing bacteria essentially take nitrogen from the atmosphere and turn it into nitrogen that is readily accessible to plants. So legumes have this symbiotic relationship with those bacteria. And leafy plants, like let's say spinach, they tend to thrive in an environment rich in nitrogen. So the legumes will be putting the nitrogen that would be necessary for the leafy plants to thrive, essentially. And that's essentially how crop rotation works in terms of fertilizing the soil. There's also things like pest control and disease control. Like if we have like the larvae or eggs of a pest and they thrive in legumes and you put a leafy vegetable there instead, we know that the disease can no longer thrive there. Next slide, please. Okay, so Winogradsky columns. So like we were talking about earlier, Winogradsky columns are a microbial ecosystem. And for any system to work, there has to be some sort of symbiotic relationships involved. And I underline model microbial ecosystems. I'm pretty sure you guys have heard, well, I imagine some of you have heard about model organisms or model systems in science. And could anyone share what their understanding of that is?
So I know for like model organisms are like picked because they're ideal for I guess showing or even testing for like certain outcomes, because either they've been like I know like either like certain cycles that they do or like their DNA or something has been like decoded, so it's been like really simple to like figure out how they work. So I think that's like, I guess like kind of like why they use them and use them and say that they're model organisms, because they're so easy to use. But I don't know about the other part of it.
So yeah, exactly. So a model organism is something that is extensively studied. So and it's easy to manipulate, as Juan was saying. Am I saying it correctly? Yeah, they're easy to manipulate. They're easy to maintain, and that's why they're used by scientists, right? Can anyone think of an example of a model organism? Go ahead.
A sea fern, but I don't know the actual name. But it's like I know sea fern is it's like citrus something, but it's a something fern. So that's all I know about it. I used it when I was in school.
Okay. Okay. Someone said mice, E. coli. Yes. How do you pronounce that one with the C? Miranda, could you help me pronounce that?
Yeah, sorry. I also used it. I think it's Ceratopterus ricardii, sea fern.
Oh, okay.
It's really
Thank
cool.
you. Yeah.
Yeah, that's really cool. I have never heard anyone else say they've used it, so it's really cool.
Yeah, I was like, for my school, not to run a rant, but for my school, we grew one and we had to like map out the entire life cycle and write lab report about it. And then like the teacher was nice enough, if he wanted to, he gave us like a seedling and let us keep it. So I still have mine somehow.
That's amazing. Yeah, awesome.
How did you use this, Miranda?
I'm sorry.
How did you, in what way did you use your seed?
I had a teacher, a professor that worked on it, on studying how it develops in oxen, how hormones are transported through at early development, early stage development. So I used to take lots of pictures of it, but I didn't know a lot about it back then either. So it's beautiful under a microscope.
I learned something today. Thank you, guys. Okay, so yeah.
But it is really fast. It grows fast. So model organism.
Exactly. That's another characteristic. They grow really fast. Okay, next slide, please. Okay. So we'll briefly just talk about Sergei Winogradsky. He was a founder of modern microbiology, the first microbial ecologist. He was born in Russia, and wrote on the first known form of chemoautotrophy. So this is a picture of him, and behind him, his background essentially, those are Winogradsky columns. Next slide, please. Okay, so why were these columns invented? They were invented as a way to enrich microbes from sediments and from soil. So when we talk about, when they're saying enriching microbes from sediments and soil, we're essentially creating an environment that allows for optimal growth. So let's say I have a cup of soil from my backyard, and I just examine it to see how much bacteria is there. There's gonna be like 10,000, let's just say 10,000. And then in the Winogradsky column, after we've cultivated it, there might be like 100,000. So the Winogradsky column is a way to ensure we have a large population of the bacteria so that we can study them. And it reduces certain selective pressures that the bacteria would be exposed to outside, because it's the best environment for the growth of these microbes. Next slide, please. And this is the stratification or zonation layers of the column. So when we put our columns together, we will mix the soil with egg yolks, which is a source of sulfur, and paper clippings, which is a source of carbon. And that will be at the bottom of the column, the sulfate-reducing bacteria. And then as you go up, you'll see bacteria that, okay, so at the bottom we'll have the highest level of sulfur. Then we'll have bacteria right above it that thrives in a sulfur-rich environment. Then bacteria just above that require less sulfur to grow. And as you go up, less sulfur is needed. As you go up, even less. So that's what allows for the stratification in this column. If you look at the picture here, you'll see the sulfide concentration is high at the bottom, and as you go up, it's lower. You can see the inverse for oxygen. So we have more oxygen at the top, and as you go down, there's less. So that tells us that the bacteria found at the top thrives in an oxygen-rich environment, whereas the bacteria at the bottom doesn't. Next slide, please. And we will look at an interactive Winogradsky column. So at the top here, we have cyanobacteria. Cyanobacteria, I've never heard anyone pronounce it, so I might be mispronouncing it. So yeah, and that's where photosynthesis happens, because that has the most sunlight. So those bacteria, we know they go through photosynthesis, whereas let's say the bacteria at the bottom, they go through more of a chemical. They use more they utilize chemicals to obtain their food. And we can go down to the heterotrophic bacteria. And on the left, we'll have a little description on it, but you can see what the actual bacteria kind of looks like. And then we have iron oxidizing bacteria. Could you click background on the top right for me, please? There's scroll down a little bit. There's an interactive, a little bit more. A little more. Yeah. So this animation just shows us what reduction and oxidation means. So these two atoms interact with each other. One of them loses its electrons, the other gains, and the one that loses is oxidized. The one that gains is reduced. But it's just to give you guys an idea of what reducing means when you look at the column, what oxidization means when you look at the column. Can we go back to interactive column, please? Thank you. Yeah, so yeah, we're just going down just to look at what the bacteria looks like essentially. I loved organic chemistry, actually. I think it's the best chemistry. Yeah, so yeah, we'll just go back to the slides and I think we can into. I think we're going to the, okay, yeah, bioart. So essentially we're trying to use this Winogradsky as an example of bioart, which is essentially an art practice where humans use live tissues, bacteria, living organisms, and live processes. I'm no expert at this at all. So what causes the different colors? So I'd imagine it's the reaction that those bacteria go through. So like the bacteria that's closer to the sulfur, they would have a different color than the ones that are further from the sulfur because they use different chemicals to obtain their energy. Next slide, please. And these are bioart examples. The one on the left, that is fluorescent proteins done in Dr. Chian's lab. I'm not sure where that is, but I know he won a Nobel Prize in chemistry in 2008. And on the right, that's a bioartist, Eduardo Katz. He essentially injected the ovum of a rabbit with a fluorescent substance, which caused the offspring to be fluorescent. So these are some columns that I tried. Oh, wait, maybe I need one. Yeah, no, can it be? Okay, maybe this. So this is like the control. This one just has sulfur, so the egg yolk. Can you see the color difference? I'm not sure. Anyone? Can you see the color difference in these two? Yeah, no?
It's not that clear to me.
Okay. Okay, maybe I'll just, no, that's too, okay.
I think we need the light on the camera side, like, you know what I mean?
Got it. Can you see now a little bit of color difference?
Yeah.
Okay, so this here is the control. This one has sulfur. This one has just carbon. This soil sample was taken from my backyard. Okay, so I covered these a little too tight. Air started to build up in them, like the bottom would round off, so it started toppling over. And this one, if you can see, the cover broke off. I don't know when. I just noticed it this afternoon, so it was pretty recent because I check them daily. And this sample, I'm not sure if you can see this one. This one I think has done a little better. I'm not sure where this one is from. Someone delivered a shed to us, and it came whole. It was already assembled, came on a truck, and then they needed a tiny vehicle that's on the truck to remove it, to bring it around. So that mud from where that that vehicle was before was preserved in the wheels. So it made a mess, and I was able to scoop it up and use it. Okay, so I'll remove these for now, and then we'll make our own columns. Okay, so, oh, I think I should share the document so everyone could see the instructions and follow along if they'd like. Oh, sorry, everyone. There we go. Okay. So this bucket here has our soil, our mud. I have a cup and half of a cup. I'm gonna put them, I'm essentially gonna portion them out and then start mixing them. So I need a cup and a half for each source, for each sample. So I'm going to have sulfur by itself, which is the egg yolk. I'm gonna have carbon, egg yolks here. I'm gonna have a carbon source, which is the chipped up paper. And then I'm gonna combine them, and then we're just gonna have the control where we're not adding anything. We just want to make a comparison. Okay. So this is, okay, this is gonna make a mix. So I'm just putting this here. This is gonna be our mixing container. I essentially used bottles, and I didn't have a funnel, so I just cut the top of the bottles and invert it in here so that I could use that as a funnel. If you can see, yeah.
So, Aransha, what is in the white bucket exactly?
Oh, so this is just soil. I can show you. This is just our soil sample. Yeah, so we're gonna mix our carbon in here. But first I'm just gonna pick out a cup for each, and then we're gonna do the mixing. It's going to be for carbon and sulfur. I already have these labeled. You don't want any contamination. Just throwing them in there. This is for a sulfur source, which would be our egg yolks. And for the control, I'm just gonna throw it right in its original container that we're gonna store the entire experiment in. And we're finished with the control. I'm gonna have a lot of cleaning to do tonight. Okay, so this one is where we're gonna put our carbon and sulfur. So our carbon source is paper. We have, that's cellulose. Throw that in there. And I already poured out the egg whites, just the egg yolks are in here. So I'm just gonna throw it in there and mix it in. So for the last experiment, I used tap water. I don't know if it affected the experiment or not. Maybe the experiment needs more time. I don't know, but you could try using distilled water, or let's say you get your soil sediment from a pond. You can use the water from the pond. Then here we have just carbon. So I'm going to take another handful of paper and just throw it in there. Mix it in. And this one is just sulfur. So what are we using for sulfur? Does anyone remember?
Egg yolks.
Yay! Thank you. Okay. So this is our egg yolk. I got a piece of paper in there, so I'll take it out. And just throw it in there. Mix it in. Okay. You guys are funny in the chat. Yeah, so we mix them all separately in their own containers. Then I'm going to add some water. I should have brought. Give me 1 second. So I'm using Poland Spring. Adding some water in there. I'm not using tap water this time. Hopefully there's a different outcome. So this is our funnel and container for the carbon source, for the column that will have our carbon only. So this is the container for the carbon only. I'm gonna just throw it in there. We have our funnel, and it's making a mess. I think I'm doing a good job, but I'm making a mess. Okay, this all doesn't want to go in there. I probably should have mixed it better. So I'm just going to pour it again. Mix it in. So that will loosen up the soil that's settled at the bottom. Try pouring it again. Oh no, it stopped.
Wait, Aransha, I'm a little bit unclear what you're doing by
Oh,
pouring it. What is the? Are you putting it through a funnel, or are you just like pouring it to mix? Sorry, I was just a little bit.
okay. So I'm sorry, guys. I am pouring it through a funnel. Well, the funnel that I made by cutting the bottom. So I'll be more clear with that the next one. So this is our carbon source. Just going to try to scrape some of it out. Can you see?
Yeah.
Okay. So this is your carbon source. Then this will be our carbon and sulfur. This one is just sulfur. This one is carbon and sulfur. So all of these are labeled already. So I'm just mixing it again, just to loosen up the soil that has settled at the bottom. Maybe I should move this out of the way so you can see. Okay, so funnel. You guys get to see that? Yeah? Okay, just gonna let this drip. And finally, we have just our sulfur source. So this one might be a little easier because there's no paper involved. So I have another spoon. Just mix it up. Yeah, this one is a little easier. Maybe I should have, so I used a shredder to shred these, and maybe I could have done something else that would have made it a little smaller, which would have allowed it to go through the funnel easier. But the egg yolk and the soil is much easier to pour. And then you can use plastic wrap to cover these up, but I didn't have plastic wrap, so I improvised, so just cut these Ziploc bags. I'm going to place it on top of the container, like so. And I'm going to add a rubber band on it to keep it, to hold it in place. And
There's a question in the chat, Aransha, that's, is there a reason one needs to pour from one bottle into another instead of just leaving mixture in original bottle and mixing vigorously?
When I did it, I just essentially put it directly into the bottle, but this instruction has to pour it over. I'm not really sure. I don't know if there's a reason. I'm just, following what they have here.
And then there's another question, would any paper work, or does it have to be newsprint?
Any paper would work, yes. Any paper would work. So the paper serves as a source of carbon through cellulose, so any paper would be fine.
And one more question. It's in soil that is agriculturally intervened, would there be less layers or less dense layers?
I am not sure. That's a good question. Maybe we could try that out. I don't know. Okay, so removing these, I'm removing the corners. And I'm just, oh, that's fine. And I'm just adding the plastic on top, like so. Putting a rubber band to hold it in place. And remember, we don't need anything to cover, anything that's too tight to cover this. We'll lose whatever is covering it. This is our sulfur source. And with the sulfur source, you almost immediately see two layers because of the egg yolk. Can you guys see it? Like one is darker down here and one is a little lighter. Again, just covering it. And we have our control. No. And so my windowsill is right there. That's where these will live for the next few weeks, about 4 to 8 weeks. And which bacteria do you guys think we expect to see at the top? What will they make the most use of? Anyone? Anyone?
Cyanobacteria is coming up in the chat.
Oh, okay, thank you. Okay, guys. Yes, why is that so? Yes, exactly. I'm bringing the lights back. So that's not disappearing.